Hi, everyone. Welcome. Today, we're going to hear from Andrew Jackson, who's a professor of neural interface at Newcastle University. Andrew trained with Roger Lemon at University College London, and then at FETS at University of Washington, where he did a fabulous experiment where they recorded from one region of the cerebellum in the macaque, and then they stimulated using that input in another region, illustrating the effects of plasticity. He, as I mentioned, is now a professor at Newcastle University, and he is really this imaginative scientist who not only does basic science, but was also interested in using ideas about stimulation to help people that have neurological disorders. So, Andrew, thank you so much for being with us today.
Yeah, well, thank you very much for the invitation. Lovely to speak to you guys about this. So, I have two. So I have should disclose that I'm involved in two very early stage neurotechnology spinouts, but they don't have any relevance to the contents of this presentation. More importantly, I should acknowledge that this is largely the work of Wei Zhu, who's now at the University of Edinburgh, but worked with me on this in Newcastle. And I'm going to tell you mostly about some monkey data about communication between the cerebellum and the neocortex in sleep. I'll talk to you a little bit about a computational hypothesis we have and some human data that maybe backs that up. But the monkey work is predominantly published now, and the references down here. So the setup for this, I think, is really quite straightforward. I mean, we know that the cerebellum is important for learning procedural skills like motor sequence tasks, and this just shows some fMRI data from humans learning a motor sequence task over several days, and it's showing that the pattern of activity in the bold signal progressively shifts from more in the cerebellar cortex, then the cerebellar nucleus to the neocortex after a few days of learning this task. We also know that sleep contributes to the consolidation, and actually not just consolidation, but offline improvements in the performance of procedural tasks like sequence learning. So this is a very similar task. This is humans learning the sequence over 2 days. And what you can see is that at the start of the second day of training, that the subjects are quickly exceeding where they had sort of plateaued in the day one learning task. And interestingly, the amount of this offline gain during sleep is proportional to the amount of sleep spindle activity. These are fast sort of 10 hertz oscillations seen in the neocortex during stage 2 sleep predominantly. So it seems the more sleep spindle activity there is, the more offline gain of skill during the night. And this sort of, to me, leads to a relatively obvious question then. So what happens in the cerebellum during sleep? And when I first got interested in this, I figured I could just go and ask someone. So I asked all of the human sleep people I knew, and they said, oh, well, we don't really know because it's very hard to record an EEG signal from the cerebellum. Cerebellum has this sort of very folded, convoluted architecture, which means that you don't get a large population field potential summing at the surface of the skull. So then I went to talk to the animal sleep people that I knew, and I said, what happens in the cerebellum? And they said, oh, we know where the cerebellum is. That's where we put our ground screw, because the human guys told us nothing happened there. So then I thought, well, maybe I can find this in the literature. But I was so surprised to find there really wasn't very much on this topic in the literature. And indeed, I came across this paper from Christa Zhu's group, where they described the cerebellum as an uncharted land in sleep research. And they pointed out that we really know very little about cerebellar processing in sleep or cerebello-cerebro interactions in sleep. And yeah, they described this as the sleeping cerebella as an enigma in the field of neuroscience. And I kind of like the idea of uncharted lands, and we thought we could kind of venture into them a little bit, because in my lab we had for some years been using this wearable battery-powered electronics to get recordings from the brain over 24-hour periods in these unrestrained macaque monkeys, including while they were asleep. And so we had mostly been doing work in the neocortex and primary motor cortex, where we use these arrays of movable microwire electrodes. This goes back to my days with Eb Fetz in Seattle. But the nice thing about these electrodes is they can be positioned to record from neurons. We get very stable recordings over multiple days. But then if we lose the neurons, we can move the electrodes up and down to find new cells. We had to adapt this design slightly to record from the cerebellum. These flexible microwire electrodes wouldn't get through the tentorium on their own, so we put them inside a guide needle. And then on the outside of the guide needle, we attach a linear electrode array, which is useful in surgery for telling when we've penetrated through the tentorium with the guide needle. We can then lower the electrodes, the microwire electrodes, down out of the guide needle and try and record from mostly Purkinje cells, we think. The linear array also gives us a depth profile of the local field potential down to the nucleus. And we targeted in the first set of animals, we targeted areas of the cerebellum and the neocortex that we know from the anatomical work of Peter Strick and colleagues are sort of bidirectionally anatomically connected. So we implanted an array in primary motor cortex and an array on the contralateral cerebellum. And like I say, all the data from 24 hours of recording gets stored to a flash memory card in the implant, and we can then take that out the next day, change the battery, and do continuous recordings of the same cells. We can typically record cells in the cerebellum. They're less stable than the neocortex, but we can typically record the same cell for a couple of days. Okay. So then, this is a sort of example of an M1 cerebellum recording. We're recording here during the night. And so the first thing to say is that, well, the cerebellum is active during sleep. And indeed, if we look at the firing rate of, in red here, a cerebellar neuron, in black here, a neocortical neuron, you can see that the firing rate is fluctuating up and down on a period of about an hour, which is the known sleep cycle in monkeys. So we have periods of relatively high activity in the neocortex and in the cerebellum, relative periods of quiescence. And that's associated in the motor cortex with this sort of alternating pattern between power in the field potential in this higher sort of 10 to 15 hertz spindle band, alternating with more power in the sort of slow wave delta band as the monkey goes through sleep cycles from light to deep sleep. And although the field potential is certainly sort of weaker in the cerebellum, we can actually see a similar pattern of alternation between high frequency, relatively high 10 to 15 hertz frequencies and then lower slow wave frequencies also in the cerebellum. So actually, in terms of both the firing rates of neurons and the sort of broad architecture of oscillations, there seems to be quite a lot of similarities between what's going on in the cerebellum and what's going on in the neocortex. And it goes a bit deeper than that, because actually these oscillations are kind of coupled. So here you can see some in deep sleep, slow wave oscillations in primary motor cortex and corresponding slow wave oscillations at the same time in the cerebellum. Here in light sleep, you can see this higher frequency spindle oscillation in primary motor cortex. So again, these spindles are described as waxing and waning oscillations lasting kind of a few seconds at 10 to 15 hertz. And we see here a spindle in primary motor cortex, but there's also a spindle oscillation going on at the same time in the cerebellum. And indeed, when we do cross-correlation between field potentials, and I should say the field potentials are locally referenced. So this is a locally referenced differential in M1 and a differential in the cerebellum. But when we do a cross correlation between them, we see that both the slow sort of delta and slow oscillation frequencies are correlated, and also these higher frequency spindle oscillations seem to be correlated between the neocortex and the cerebellar cortex. So these oscillations are coupled. The next question was, is there any directionality of that coupling? Is one oscillation driving the other? And to look at this, we used directed coherence, which is a frequency domain version of Granger causality. And effectively, it's saying to what extent is the past history of the signal in one area predictive of the future evolution of the signal in the other area based on sort of autoregressive models. And this result, actually, I consider this to be one of the sort of more surprising results that I've got in my career, and I'll sort of say why. But what we found when we looked at the directed coherence was we saw a very nice dissociation between these two frequency bands. So when we looked in the direction from the neocortex to the cerebellum, we found that the directed coherence was dominated by the low frequency, sort of slow wave and delta frequencies. Whereas when we looked at the direction from the cerebellum to the neocortex, that's when we saw this directed coherence at the 10 to 15 hertz spindle band. And this is a sort of surprise because about the only thing that anyone agrees upon in relation to sleep spindles is that they come from the thalamus and the thalamic reticular nucleus. In fact, that's the first line of the Wikipedia entry on sleep spindles. But given the sort of known anatomy of the cerebro-thalamo-cortical connections, it's very hard to see how a spindle oscillation arising in the thalamus could be seen first in the cerebellum and then in the motor cortex, because there's not really a direct connection from the cerebellum to the neocortex that doesn't go through the thalamus. Now it's worth noting that the original evidence for why sleep spindles emerged in the thalamus was sort of early work by Steriada, who showed that if you lesion the motor cortex or lesion the neocortex, spindles remain in the thalamus, but that if you lesion the thalamus, you lose the spindles from the neocortex. But of course, lesioning the thalamus, you're also disrupting the input from the cerebellum. But anyway, extraordinary claims require extraordinary evidence. So in another set of animals, we also put electrodes into the thalamus to record during the night, and we made sure we were in the right place because we could see antidromic fields on the electrodes that were going down into the cerebellar nucleus, and we could get orthodromic fields from stimulating up in the neocortex. So we're pretty sure we had our electrodes in the right place. And then we again repeated the directed coherence analysis. And what I'm showing you here is directed coherence between the thalamus and the cerebellar cortex. And you can see again, we don't see any spindle directed causality from the thalamus to the cerebellum, but when we look from the cerebellum to the thalamus, we do see this peak at spindle frequency. So it really does seem consistent with the idea that these spindle oscillations in the cerebellum seem to be driving the spindle oscillations in the thalamus and the neocortex. We also did an analysis to try to look at the relative timing of the amplitude envelope of the spindle oscillations. Did one sort of start or peak slightly before another? And this is just showing you the result of cross-correlating the amplitude envelopes in the spindle band. And we find that this, indeed the amplitude of the spindle in the cerebellum seems to lead the motor cortex by about 10 milliseconds. It's quite noisy, this analysis, but on average it's about 10 milliseconds, and that seems to be made up of about 5 milliseconds from the cerebellum to the thalamus and 5 milliseconds from the thalamus to M1, which is sort of it's in the right range for these kind of conduction latencies. Now, before we completely kind of rewrite the Wikipedia entry on sleep spindle oscillations, I should tell you one more thing. I think that it's a little bit more complicated than that. I think we have evidence in our data for the fact that there may be at least 2 separate oscillating circuits. And this came about from a sort of rather more detailed analysis of the relative phase of the spindle oscillations that we were recording in the cerebellum and the neocortex at the same time. And what we noticed looking at the data is it seemed like that relative phase was changing through the spindle events, even though kind of on average it was these 2 oscillations were coupled. There seemed to be systematic variation in the relative phase. And when we looked at this, so this is just a histogram then of the instantaneous phase difference between oscillations, and this is just one session, one pair of electrodes. The instantaneous relative phase between M1 and the cerebellum, and you can see this is peaked around a preferred phase, but there's quite a kind of a width of epochs where different relative phases are being shown. And what we found is that actually what we found is that the epochs that are associated with the cerebellum being relatively earlier compared to the neocortex are actually associated with times where the amplitude of the spindle oscillation in the cerebellum is increasing, and vice versa. So this plot here is showing the rate of change of the amplitude of the spindle for those epochs where the relative phase is a sort of given along the horizontal axis. So effectively what we're saying is over here these are periods where M1 the motor cortex relatively is leading in the spindle oscillation, and you can see that on average for these phase differences the rate of change of the amplitude envelope in motor cortex is positive, whereas the rate of change of the amplitude envelope for the cerebellar spindle is negative on average. And this behavior is actually very characteristic of coupled oscillators. You may have seen the sort of demonstration where you have 2 pendulums and they're attached together with a spring, right? And you start one of them going and the energy kind of propagates to the other pendulum, and then the energy propagates back. And so there are these alternating periods where one pendulum is driving the other. And that is associated with systematic differences in the phase of those coupled oscillators. And so we actually built a very simple model that captured this behavior, where we had 2 oscillators. Here I've labeled them M1 and cerebellum, but there are sort of caveats as to where those oscillators could be. They could also be kind of upstream and downstream of the relative areas. We have 2 oscillators here. We have coupling between them, and these oscillators are excited by both a shared common input and some independent common input. And I think this, if I remember rightly, this model has only I think 8 parameters that basically capture these various coupling strengths, and then we fit the model to all the data from a night's sleep. But then once we've fitted these connection weights, we can then just put white noise input into this model, and we can generate new data based on just those 8 different parameters. And when we do that, we find that this model does indeed generate sort of things that look a lot like the spindles in our data, but moreover, this model can recapture, can recapitulate that behavior of this sort of waxing and waning amplitudes associated with the systematically associated with the relative phase of the oscillations. And we can't get this behavior if we take out any of these couplings. So if we only have one oscillator in the model, we can't produce this behavior. And so I think this suggests that there are at least 2 oscillating circuits that are coupled with each other. And we would suggest that maybe one of these is the sort of well-known oscillatory circuits in the thalamus, but there may be additionally oscillatory circuits oscillatory networks in the cerebellum or upstream of the cerebellum, which is sort of coupling in and driving that thalamic spindle during the night. And the net effect of this is a sort of relay of causally propagating information from the cerebellum to the neocortex during the night, during these sleep spindles. And you know, you'll remember that I said to you that the the sleep spindles that seems to be predictive of the amount of offline improvement that we get in these procedural tasks after a night's sleep. And indeed, the neocortical spindle is thought to be a very good pattern of activity for driving plasticity in neocortical circuits. Okay. So that was field potentials. What about spike activity then? So what I'm showing at the top here is spike coherence between all our pairs of neurons, separated for sleep in blue and awake in red, and for pairs of motor cortex neurons, pairs of cerebellar cortex neurons, and between area pairs. And you can see kind of actually interestingly within area, we don't see very much difference between wake and sleep in the spike coherence. But the big differences are when we look at the between area correlations. And I've zoomed in on the spindle band here, and you can see that in sleep we see an increase in spike coherence at spindle frequencies, but we see a reduction in spike coherence at these low frequencies that are kind of associated with movement. These are the sort of frequencies of dominant frequencies in the movements that the monkeys are making during the day. And this is just an example here of a cross correlation histogram for an example pair of M1 and cerebellar neurons showing this nice oscillatory spindle frequency coupling in the spikes. So, to summarize, increased low frequency coupling during waking behavior and increased spindle frequency coupling during sleep. Now, we were interested in doing a bit more to reassure ourselves that these sleep spindles in the cerebellum did indeed sort of share the expected features of what's been reported for sleep spindles in the past. So people tend to report that spindles are associated with transitions from down states to up states, which is the slow oscillation going on at lower frequencies, is this sort of transitions from quiescent down states where neurons aren't very active to up states where neurons are much more active. And that's when spindles tend to occur. And so we used some of the techniques that have been validated for human actually depth EEG recording to identify our upstates from slow oscillations in the neocortical field potential. And we also identified our spindles using the sort of the standard human definitions of what counts as the spindle. And we found that indeed the spindles did indeed cluster around the neocortical upstates, which were actually indicated by a depth negative field potential. And then we looked at our spike to spike coherence, so the spike coherence between the cerebellum and the neocortex, with a sliding window around that upstate, and we found that indeed the upstate was also associated with increases in coherence at spindle frequencies between individual neurons in each area, and also an increase in coherence at lower frequencies, these sort of slow oscillation frequencies, where, if you remember, said the direction of causality is in the other direction from neocortex to the cerebellum. And we could also, because we're looking at firing rates, we can also just look at the firing rates on average in the two areas associated with this down to up state transition. And so what I'm showing here in green, and these are different axes, but I think it's sort of instructive to actually scale the axes the same here. So in green, I'm showing the average firing rate in the motor cortex relative to the upstate, and you can see that the firing rates are maximal at the time identified from the field potential negativity as the upstate, and there's a downstate sort of preceding it, relatively lower firing rate. You see the average activity in the cerebellum follows a very similar profile, but there's actually a time lag here. So these low frequencies associated with the transitions from down to up states, it seems that the motor cortex is indeed leading and the cerebellum is following, and that's consistent with that directionality that we saw from motor cortex to the cerebellum at low frequencies. But one of the things that's sort of interesting about this is this sort of this pattern. Well, okay, and then I should say, so is it just that the motor cortex always leads the cerebellum? But actually it's not the case. So if we look during the day and during movement, and this was actually a trained task that we had animals making movements, you can actually see that it's sort of low frequencies, the profile, the frequency content is kind of similar, right? There's a sort of rise and fall in activity as animals here were making wrist movements. But you can see that in this case, the cerebellum and the motor cortex are on average sort of peaking in their activity at the same time related to movement. Ignore the thing over to the right. This is just the second part of the task that seemed to activate the cerebellum, but not the motor cortex. But at the time of the movement, there seems to be kind of roughly the same. These areas are active at roughly the same time, possibly even cerebellum a little bit in front of motor cortex, whereas during the night, it seems very much that the motor cortex is driving the onset of the upstate, and then the cerebellar activity is following. And without wanting to go into too much detail about this, we got quite interested, despite that difference, is there actually a similarity between the pattern of activity that we see in this cerebro-cerebellar network during waking and sleep? And we were interested in this because we've done sort of previous work in motor cortex showing that the pattern of activity that you see around these down to upstate transitions actually kind of resembles the pattern of activity that we see during movement in terms of the precise relationship of the cells, a little bit like replay, although I wouldn't say we have quite enough data to make claims about replay. But so we looked at this in terms of the relative order that cells fire in. So here you're seeing around the time of a movement. You can see here there's two cells, and cell 1 is firing a little bit in advance of cell 2. And then during this upstate transition, you can see again, there's this sort of transition from quiescence to high activity. And again, cell 1 seems to be active slightly before cell 2. And obviously this is an example that I pulled out. When we look across the population, it's not always as clear as that, but we find that when we characterize the relative order of cells in terms of the asymmetry of their cross-correlation histogram, we find that across the population, that is indeed correlated between waking and sleep. So if a cell tends to fire earlier during a movement relative to a second cell, it also tends to fire earlier during an upstate transition relative to the second cell. And that's true whether we take pairs of neurons in motor cortex, pairs of neurons in the cerebellum, or pairs of neurons between motor cortex and cerebellum. So I've shown you a lot of data, so I'm just going to kind of summarize this now in cartoon fashion. So what we think is going on during movement, we see relatively contemporaneous low frequency activity in the neocortex and the cerebellum, which presumably reflects the bidirectional communication that's going on between the cortex and the cerebellum to shape the kinematics of the movement. In sleep, we observe kind of population dynamics at these low frequencies that also resembles that seen during movement, but with the difference that the cerebellar activity lags behind the neocortex, and the causality is all directed from the neocortex to the cerebellum. But at the same time, we see spindle frequency coupling between the neocortex and the cerebellum. And in the case of that spindle frequency coupling, the causality is directed from the cerebellum via the thalamus to the neocortex. Okay, so I have 10 minutes left, and so I'm going to go through this next bit relatively quickly, although I'm happy to discuss it further. So this is this is an attempt to try and make sense of it, and it's an attempt to try and make sense what actually is going on during the night. And we know that, anecdotally, if you've got a problem, you should go and sleep on it. We know from various scientific studies, like this one, where people were trying to find a secret strategy hidden in a task, and this was a nice study of inference that if relative inference, so you learn that an abstract A is bigger than B, and you learn that an abstract B is bigger than C, and then you're asked, what's the relationship between A and C. In both of these tasks, after a night's sleep, people are actually better at doing the task. And I like these tasks because the discussion of these studies is virtually identical, but they both sort of say, we conclude that memory representations are restructured or kind of formed into meta-memory representations with the aim of guiding insightful behavior or flexible behavioral repertoires, right? And this is kind of a wordy description of what might be going on to learning during sleep. And I always found this slightly unsatisfying, right? Because what I want to know is, well, how are memories being restructured to facilitate behavior, right? What does that mean that the memory is being restructured? And also, crucially, why does this have to happen in sleep, right? Why don't we just learn things in the structure that facilitates behavior in the first place? And I think that there's sort of elements of what I kind of learned over the years in thinking about motor control that are potentially relevant here. So we kind of know, right, that behavior, if the aim is to facilitate behavior is a hard problem, right? And it's a hard problem because it's an inverse problem, because we're trying to find a mapping between our desired goal, which is a consequence, and we want to map that onto our necessary action, which is a cause. And I don't need to tell anyone in the room here that those kind of inverse mappings are ill-posed problems. There may be many ways to get to your desired goal. There may be no way to get to your desired goal. So in general, it's a sort of one to many or a one to none potentially problem. It's also a problem because the world provides indirect training signals. The world just tells you whether you got to your goal or not. It doesn't tell you whether there was a better way of getting to your goal, or it doesn't tell you what you should have done if you didn't get to your goal. Now, of course, we have, and so it's a sort of reinforcement learning problem, and we have all sorts of powerful methods of solving reinforcement learning problems these days, but they require lots of training data. So they are generally, very data hungry to solve these kinds of problems. Now, there's another kind of learning that we can do in a system, which is learning to predict the consequences of our actions, sort of forward models or forward predictive models. And in this case, it's a forward model because it's a prediction for it's a mapping from a cause, which is an action, to a consequence, which is the resulting state. And these kind of mappings have all sorts of advantages for learning. They're well-posed problems. Every action has a consequence. And moreover, the world provides a direct training signal because the world tells you what actually happens. And so if there's a discrepancy between what you predicted and what actually happens, the world is telling you what you should have predicted. So you can learn these problems as a supervised learning problem with a sort of error-based approach. And as a result, you can learn these kind of forward models with much less training data. But the surprising thing is that despite all these sort of advantages of learning forward models versus learning inverse problems, if you've learned an accurate forward model of the world, that's all you need in order to produce an optimal inverse controller. And many of you probably know this, that there's many different ways of inverting a forward model into an inverse model, but basically they're models of the same thing. So an accurate forward model does contain all the information about the world, that is all the information you would need to control an agent in that world. And I'm going to sort of argue that there's one particular way of inverting a forward model that might be relevant to this circuitry that we've been describing. So the idea is that we have an inverse controller potentially located in the neocortex. And generally that, during the daytime, we can be learning, we can be optimizing that controller by using reinforcement signals from the world that tell us if we reached our goal. But at the same time, we can also take an efference copy of our action, and we can learn via supervised learning to predict the consequences of our actions. And we think that may be what's one of the things that's going on in the cerebellum, right? And then the idea is that during the night, what you can do is you're disconnected from the world, but you can still generate more data to train up your inverse controller via reinforcement learning. But now that data is not real exploration in the real world. It's simulated data from sort of fictive exploration of a forward model and the forward model, the forward predictive model is telling you what would have happened if you'd done something, and you can use that information to derive a fictive reinforcement learning signal that can allow you to do more optimization of your controller offline without any real world costs of that learning. And this is an architecture that actually introduced by Sutton. It's called DynaQ, and more recent variants of it are often used to have been proposed to explain what's going on with hippocampal replay during the night. But it strikes me that at least in the context of our motor cortex and cerebellum data, we see something that is at least consistent with this, right? That our slow wave activity is looking like a kind of, it has the same structure as activity that we're seeing during a movement during the daytime. But what we're getting back from the cerebellum to the motor cortex is now some signal that's at spindle frequencies that is very effective at driving plasticity in the neocortex. And so this could be a training signal based on a forward predictive model that could be driving further plasticity to optimize the controllers during sleep. So our sort of simulation of this, this is just a kind of model here, and we're simulating an agent that has to move through 7 different states. And the agent can choose one of 2 actions at each state that takes it to another state. And the agent just has to get to a goal state with the smallest number of actions. And you can learn that with Q-learning. And it turns out, actually, it's a sort of surprisingly difficult thing for Q-learning to learn. It takes, these are action blocks of 50 moves, so it takes quite a lot of having a go at this to learn the most efficient way of getting from each state to another. But at the same time, our agent can use supervised learning to also predict where would it get to if it did a given action in each state. And as I said, that's a much easier task to learn. So actually, when we test how good is the forward predictive model doing, the forward predictive model saturates up at 100% really relatively quickly in this task. And then we can simulate sleep by a period in the middle here where we turn off the agent moving in the world, and we just generate sort of fictive goals that are turned into fictive actions that make predictions and are used to drive further reinforcement learning. And we find that this model improves its performance after a night of simulated sleep, even though it hasn't actually had any new information from the world during this period. But of course, these improvements do depend on having an accurate forward predictive model. So if we degrade the forward predictive model before this simulated night sleep of learning, we then see that we don't get the same improvement after running further controller optimization on the offline data. And so this led us to a kind of hypothesis. Well, the sort of hypothesis is that if this model is right, then offline improvements in an inverse control task should depend on the availability of accurate forward predictions at the end of day 1 learning. So we set out to test this in humans, and I'm not going to go into too many details, but the structure was exactly the same as the simulations I've just showed you. Here, each state was indicated by a kind of arbitrary circle on the screen, and the actions where people pressed a left button or a right button to move to the next state according to this abstract kind of mapping. And we did 2 types of tests. One was to test people's inverse control abilities. So we asked them to press a sequence of buttons. So we said, here's where you currently are, and here's the goal, your goal, and get there in the minimum number of buttons. And in this case, the best thing to do would be to press the left button, which takes you there, and then press the right button. So here, the best thing to do is press left, then right. And people learnt that for day 1, and this was sort of several hundred trials. And I'm showing you here, well, okay. And then what we did was, at the end of learning, we did a second test where we tested their ability to make predictions about the consequences of their actions. So in the second test, what we showed them is we gave them a current location and we gave them a sequence of actions. So we said, if you pressed left and then right, where would you get to? And they had to indicate with a mouse click which target they thought they would get to. So the control test is, can you generate actions to get you to goals? And the prediction test is, can you predict the consequences of your actions? And based on the results of the prediction test, and we didn't give them any feedback because we didn't want them to learn anything from that. We separated them into good predictors and bad predictors. And actually what I'm showing you here is two groups of subjects, good predictors and bad predictors, who've been balanced for their performance on the control task on day one, right? And then they go away for a 24-hour period and they come back and they do the control task again. And it's only in the good predictors that we see this offline improvement in performance. And we've done this, we did another study where we said, well, does it depend on time or just sleep? So we did 3 learning sessions, morning, evening, next morning, or evening, next morning, subsequent evening. And without going into too much details, we find that this relationship between how good you are at predicting and then how much offline improvement there is in a kind of inverse control task only seems to hold up for the overnight period. So we think that this may be a mechanism that is actually reliant on sleep. So I got through quite a lot there, but I got through everything I want to do. And I'm just going to leave you with this sort of pictorial summary of, in the top, the summary of the monkey electrophysiology, where we have the pattern of connections in waking and sleeping, the different frequencies we looked at, and in the bottom then our kind of speculative hypothesis about how this may actually form part of a learning mechanism for learning to predict the consequences of your actions during the daytime, and then using that knowledge in order to train up inverse controllers during the nighttime as a potential role for the cerebellum in offline learning of procedural tasks. And just acknowledge the people who did the work, and like I say, mainly Wei Zhu in my lab and funders. And thank you for your attention.
Oh, Andrew, that was just marvelous. A beautiful tour you took us on. Let me see if we have questions for Andrew. I will get started. Andrew, can you give us more information about the recordings that you did in the cerebellum? Maybe a little bit about, you mentioned some spiking coherences that you measured. Do you have a sense of what neurons you might have recorded in the cerebellum?
That is a very good question, and it was obviously something that came up a lot in the review process. So I will say that we did not find as many complex or easily identified complex spikes as we were expecting, and I'm still slightly surprised by this. I think in part it's due to the very low impedance and very large recording area of these micro-Rs that we use. And I would say there's potentially a sort of trade-off between the kind of the quality of the recording that you'll get with a sharp high impedance small area electrode versus whether you then can record that cell for a day or two while monkeys are jumping around in unrestrained behavior. But so that's my hypothesis as to why we didn't see as many complex spikes as we wanted to. And obviously, if you want to really make absolutely sure that these are Purkinje cells, simple spikes, you would want to associate that with associated complex spikes. Now, we did find some complex spikes, and when we looked at it, we found evidence for simple spike pausing after the complex spike. And what we also looked at then was various other characteristics of the neurons. So all the sort of things that we could look at, we looked at kind of spike width and firing rates and regularity of firing and that sort of thing. And what we found was that there was no difference in those metrics between the kind of positively identified simple spikes and the majority of the rest of our population. Where there was a difference between those metrics for the cerebellum versus the pyramidal neurons in the neocortex. So on those sort of indirect metrics, we think that the majority of the neurons we were recording from were Purkinje cell simple spikes. But I would say that it falls some way short of the kind of gold standard, or the level of sophistication that cerebellar physiologists usually are able to use for kind of identifying cell types. And I think that's partly the price that you have to pay for using an electrode technology that then allows you to record for 24 hours at a time.
Wonderful, wonderful. Any other questions for Andrew? Yes, Rich, please.
Hi, Andrew. Nice talk to hear. We once did an experiment where we looked at sequence learning in individuals with cerebellar degeneration, and we actually found this dissociation between when they actually were reaching to lights, so they could reach directly to where the target was. They showed normal learning. It was only when they had to do some like symbolic transformation, like they had to say, you know, red was the key on the left. So if it was like spatially cued directly and you reach for the location, they showed normal sequence learning. But when they had to do that transformation, they didn't. And our argument there was, at least in the early stages of learning, the cerebellum, at least in these individuals, they were only affected when they had to sort of keep track of the stimulus response mapping, kind of the vertical associations, but not informing the sequential ones. And, you know, I suspect that, you know, that's the kind of thing that dominates a lot of the early stages of sequence learning. You know, it might be a very different story when you do lots of practice. And I didn't know how, with your sorts of thinking about this in the model, what you really see as the, you know, is the role of the cerebellum just in general about doing these sort of predictions, or is there some special part of sequence learning that you'd expect the cerebellum to be important for?
So yeah, no, it's a great question, and I mean, I think that the task that we used, and I would say that, you know, there are kind of two parts to that talk that I just gave, and whether the cerebellum is the kind of common link or not, it still remains to be seen, right? I mean, a lot of people would argue that the task that I showed you was a navigation task, and it's a hippocampal task, right? And so why is he talking about a hippocampal navigation task just after talking about kind of the cerebellar electrophysiology, right? And I think that, you know, we probably, just as it's worth, I think that there's not enough people studying the communication between the cerebellum and the neocortex, but I also don't think there's enough people studying the communication between the cerebellum and the hippocampus, right? I would say that, you know, in both cases, the brain areas talk to each other much more than the people studying the reflective brain areas talk to each other. But so I think it's an open question as to, you know, particular tasks like sequence learning, say, you know, the extent to which it's really going on in the cerebellum, or you know, there's a sort of memory component that relies on the hippocampus, and certainly I think there are arguments for deficits from injury to both areas. What I think is maybe the common thread is that I think if you look at the kind of emerging way people are thinking about the hippocampus now, it's as the hippocampus as a predictive model of you know, where I'm going to get to if I go you know, down the hall, or I go down this corridor in a maze. I think what's possibly different between the kind of hippocampal predictive models that say the rodent people tend to think about versus the cerebellar forward models that we think about in the motor control is that for getting around in a sort of virtual, even abstract state space, it doesn't really matter how you get from one place to the other. So the hippocampal prediction for you know, where I end up if I go down the corridor doesn't really depend if I walk down the corridor, or if I bicycle down the corridor, or I drive down the corridor, right? Whereas actually behavior involves just more than sort of getting around in a sort of state map like that. It actually involves generating the specific movements to get me around there. So I think that, you know, I don't have data to answer this, and it sounds I should certainly look into your data with the sequence learning task. But I wonder if something about the hippocampus is representing a kind of more general sort of the sort of structure of the task in terms of kind of sequences like that, whereas the cerebellum is actually involved in much more the actual actions that get you between sort of states on that sequence. And it may be that for you know, some types of sequence tasks, one is more important than the other, or one is learned earlier and the other is learned later. But actually, I think both are important if you want to perform one of these tasks.
Yeah.
I don't know if that really made a lot of sense.
No, I mean, that would kind of, the one thing I'll point out is that kind of picture might be a little backwards from the imaging study you started with, right? Because that kind of gave the picture of the cerebellum early in learning and not late, whereas I think sort of the notion of going from the abstract to the more movement specific is kind of the reverse direction, where the cerebellum might have a role more in late learning than early learning.
Well, except that I suppose what I'm arguing is that everything, I mean, and this is also obviously a massive simplification, but I guess I'm arguing that everything ends up in the neocortex after after the offline consolidation. So in a way, it's a very similar kind of argument to why, where the memory representations shift from the hippocampus up to the neocortex. So it would be this continual, and it is the sort of speculative hypothesis that's sort of simplistic, but sort of gradually what you're doing. So the reason why the memory is being restructured is the memory is being restructured from a predictive cause to consequence representation to a kind of control scheme, a kind of an action that will get me the consequence that I want, and progressive, and that is represented in the sort of neocortex more. So there's a sort of shift from relying on predictions early in learning to just generating in a sort of feed forward way your action later in learning, which would be why it tends to shift from these predictive areas, both in the hippocampus and the cerebellum, to the neocortex after a few sessions.
Yeah. Okay, thank you.
Andrew, there are a couple of questions on the chat. I wonder if you can open it. There's one by Maria asking you about TMS and its effects on cerebellar inhibition. I wonder if you can comment on that.
So I think the question is about the timing, I think, and the argument is, I think that if you stimulate the cerebellum and then 5 milliseconds later you stimulate motor cortex and you see presumably an inhibition of the MEP, I think the question is 5 milliseconds a reasonable transmission latency? And I guess I would argue that I think it is, right? So what we tend to see traditionally is, if you look at time lags with kind of natural sort of spontaneous activity, you always tend to see those time lags as sort of slightly shorter than what you see when you deliver a kind of big stimulus like TMS. And people argue about whether that is because you're sort of preferentially activating the larger diameter fibers, and you get a kind of, certainly an MEP from motor cortex to the muscle is sort of right on the fastest edge of what you then sort of measure with, say, spike trigger averaging. So we got a kind of 10 millisecond latency from our envelopes. We also did some stimulation, and I would have to sort of remind, we've done this also. I think Lee Miller did this a while ago in monkeys as well, of sort of stimulating in the cerebellum, recording in motor cortex, stimulating in motor cortex, recording in the cerebellum, looking at those kind of time lags. But actually, I think yeah, 5 or 10 milliseconds is the sort of ballpark you would expect for the sort of straight conduction time from the areas. You obviously have to kind of think about what the effect of the various inhibitory connections in that pathway are going to be. I think the harder one to kind of explain is when we're going the other way, when we look at the latency between from motor cortex to cerebellum, and that was more like about 100 milliseconds in our data in terms of the slower firing rate profiles. So that's a sort of latency that is a little bit hard to explain as a kind of as a direct conduction time latency, and must have more to do with the dynamics of the area. So I hope I understood that question right. And Yuliu said how to detect spikes using spike sorter threshold trigger. So the spike detection in this case was all done offline. So we record the raw data at I think 20 kilosamples per second onto the flash memory card. Back in the early days when I started doing this with electronics, we had to do spike sorting on the device because the biggest flash memory card chip you could buy back in the early 2000s was sort of 8 megabytes of memory, and you couldn't get any kind of length of raw data. But one of the great things about doing this in parallel with the advances of consumer electronics is it's now very easy to get sort of 128 256 gigabyte flash cards, and so it's now possible to stream everything raw. So we did not use the, although I want to sort of try to use the very nice spike sorting software that I believe that you're developing, but we used WaveClass, I think, for our spike sorting.
Sounds good, Andrew. I guess ask if are there any other questions for Andrew? I love that last experiment where you queried whether they had a good idea, but they had a forward model by asking them what would be the consequences, and that result that people who had a better forward model seem to learn more overnight. That's really striking.
Yeah, I mean, I should sort of say that in presenting that, I skipped over a slight. The two are not completely orthogonal, right? So it's not like how good you are at prediction is uncorrelated from how good you are at control at the end of day one. So I showed you a subset of subjects that have been balanced. We obviously also did a kind of multiple regression partialing out the effect on. So it's not that they're completely uncorrelated, but it's surprising actually that people can get quite good at the task and then not be able to answer that second question, and vice versa. And I think that is to some extent inter-individual differences, right? Is how they learnt it. But it would be nice. What I'd like to do is to think about ways to be able to control that rather than just use that variability between different people. It would be nice to think about ways that we could train people on tasks that would kind of bias them towards at the same time learning a predictive model or not learning a predictive model. But I haven't yet come up with kind of clever ways of doing that yet.
Yeah, no, that's fascinating. Are there any other comments for Andrew? All right. Thank you, everyone. Have a wonderful day.
Thank you very much for the invitation and taking time to listen.